Hello, you're welcome to our lectures on biological techniques. Today's lecture will be a continuation of an earlier lecture we had on the compound optical or light microscope. We had earlier discussed the different types of optical microscopes, 8 types. And in our earlier discussion, we had established that the light microscopes, the types of light microscopes include the bright field microscope, the dark field microscope, the stereoscopic or dissecting microscope, the confocal laser scaling microscope, the fluorescent microscope, the phase contrast microscope, the differential interference microscope, as well as the inverted microscopes. We have discussed about the bright field microscopes, the dark field, and the stereoscopic microscopes earlier. So we're going to continue with the confocal laser microscope. It's a type of the optical or light microscopes that we have discussed earlier. So what is the confocal laser scanning microscope? The confocal scanning microscope is quite different from the stereo and compound light microscope. Unlike the compound and stereo microscope, these devices are reserved for research organizations. They are reserved for research organizations. They're able to scan a sample also in depth. They have the capability to scan a sample in depth. A computer is then able to assemble the data to make a 3D image. Now, this is a confocal microscope, a confocal laser scanning microscope. Here, you can see that it's very sophisticated, having a computer and other digital aspects or parts of the microscope. It's able to penetrate or to see deeper parts of the organism that's being studied. Now, what are the uses of the confocal scanning microscope? In a confocal laser scanning microscope, a laser beam passes through a light source aperture and then is focused by an objective lens into a small, ideally diffracted, limited focal volume within or on the surface of a specimen. The confocal scanning microscope has the following uses. The first use of the confocal scanning microscope is that it is widely used in numerous biological science disciplines, from cell biology and genetics to microbiology and developmental biology. It is useful in most aspects of biological sciences. It's used in cell biology, genetics, microbiology, and developmental biology. It is also used in quantum optics and nanocrystal imaging and spectroscopy. So it's a very sophisticated device that's used in various fields. Clinically, confocal laser scanning microscope is used in the evaluation of various eye diseases and is particularly useful for imaging, qualitative analysis, and a quantification of endothelial cells of the cornea of the eye. So it's basically used also in the hospitals. It's used for the study of various diseases of the eye. It is used for localizing and identifying the presence of filamentary fungal elements in the corneal stroma in cases of keratoconus, enabling rapid diagnosis and thereby early institution of definitive therapy. Now, having discussed about the confocal laser scanning microscope, we want to now look at the fluorescent microscope. The fluorescent microscope uses emitted light to create images. In fluorescent microscope, many wavelengths of light ranging from the ultraviolet to the visible can be used to cause samples to fluorescence to allow viewing by eye or with the use of specifically sensitive cameras. This is the fluorescent microscope, and the majority of fluorescent microscopes, especially those used in the life sciences, are of epifluorescence design. Light of excitation wavelength is focused on the specimen through the objective lens. The fluorescence emitted by the specimen is focused to the detector by the same objective that is used for the excitation, which for greatest sensitivity will have a very high numerical aperture. Now here's the parts of the fluorescence microscope, having the eyepiece and the observation tube, the tube lens, the field diaphragm, the condenser aperture diaphragm, then the mercury arc lamp, lamp house, then the frame, and many other parts. Okay, these are the parts of the fluorescence microscope. What are the uses of the fluorescence microscope? The first use is that the fluorescent microscope is one of the uses of the fluorescence microscope is fluorescence antibody staining. One of the uses of the fluorescence microscope is fluorescence antibody staining. This method is now widely used in diagnostic procedures to determine whether an antigen is present. Clinical specimens such as blood and serum are often used for the diagnostic procedures. Blood and serum are used to check for the presence of antibodies in the sample. It uses fluorescent staining, which is used in many diagnostic procedures for identifying microbes. Now I want to look at the phase contrast microscopes. The phase contrast microscope. Phase contrast microscopy is an optical microscopic technique that converts phase shifts in light passing through a transparent specimen to brightness changes in image. The phase contrast microscopy is an optical microscopy technique that converts phase shifts in light passing through a transparent specimen to brightness changes in image. It uses phase shifts. It uses phase shifts. The light basically it's produced in phases. It's produced in phases and it passes through transparent organisms. The brightness changes and the image, because of the phase shift, the light is not produced once, it's produced brightness and darkness like that. So as it produces such phase shifts, it enables to, it uses such a mechanism to produce an image. It is an ordinary bright field microscope with two additional plates. The phase contrast microscope is the bright field microscope, but it has two additional plates, namely annular diaphragm and phase shift plates, which enables the formation of rings to be phase shift with respect to others. Now, this is the phase contrast microscope, just as it has been said. It is the normal optical microscope, but it has some additions, and those additions are the annular diaphragm and the phase shift plates that are added to create a contrast in the phases. Now here is also another image of the phase contrast microscope. Here we have the annular ring that produces phase shifts in the light that's been produced. The phase shift that is being produced. Okay, so then it also have a phase plate in the objective lenses. What are the uses of phase contrast microscopes? Phase contrast microscopy is particularly important in biology. It is essentially useful because it enables detailed examination of the internal structures in living cells and reveals many cellular structures that are not visible with a simpler bright field microscope. It is used to view internal structures in living cells, and helps to reveal many cellular structures that cannot be seen with the bright field microscope. It is also useful for studying microbial motility, determining the shape of living cells, and detecting bacterial structures such as endospores and inclusions. Okay, it's very important in studying microbial motility, how microorganisms are able to move. And it also used to determine the shape of living cells, the various shapes of living cells, and for detecting bacterial structures such as endospores and inclusions. These are clearly visible because they have infractive indices different from that of water. The phase contrast microscopes are also widely used to study eukaryotic cells. The use of phase contrast microscope does not require the fixing or staining of the specimen. Fixing and staining the specimen may distort or kill the specimen. That's one very important feature of the phase contrast microscope. It does not require fixing or staining of the specimen. The phase contrast microscope made it possible for biologists to study living cells and how they proliferate through cell division. The proliferation of cells through cell division, how they proliferate, is also made possible with the use of the phase contrast microscope. Now, these are images that are produced from the phase contrast microscope. Now, in contrast with the bright field microscope image, it is very obvious that the phase contrast microscope produces a better and clearer image. So the same cells imaged with traditional bright field microscope on the left and the phase contrast microscope are actually at the right hand side. And it's very clear that the phase contrast microscope produces better images. The next type of optical microscope we want to look at is the differential interference contrast microscope. The differential interference contrast microscope. If the beam of light is shifted in phases by a variable amount, the system becomes a differential interference contrast microscope. If the beam of light is shifted in phases by a variable amount, it becomes a differential interference contrast microscope. This microscope makes use of the fact that combining light waves that are out of phase with each other produces interference that alters the amplitude of the light wave. Similar to the phase contrast, differential interference contrast microscopy is a contrast enhancing technique. It enhances contrast. The differential interference contrast microscope uses polarized light to convert phase delays into intensity changes. The effect is called differential because contrast is created only in neighboring areas. The contrast is created only in neighboring areas. Now, this is the picture of the differential interference contrast microscope. It has an analyzer, and it also has the objective that is called the Namaski prism, and also has a condenser like the other compound light microscope, but the condenser is called condenser Namaski prism. Then it also has a polarizer and It has a fluorescence and other features. What are the uses of the differential interference contrast microscope? One, it was once used nearly exclusively by parasitologists. This type of microscopy has increased in use because of the work currently done on the nematodes C. elegans. Interference microscopes are superb for both observation and measuring thickness of embryos within specimens with little or no contrast. The phase contrast microscope, this microscope is used to visualize live cells and living events and does not require staining. It is used to view live cells and does not require staining. Now I want to look at the inverted microscope. Inverted microscope. Now, what is an inverted microscope? An inverted microscope is a microscope with its light source and condenser on top, above the stage, pointing down, while the objectives and turret are below the stage, pointing up. It is said to be inverted because the light source and condenser are on top. The light source and condenser are on top instead of being at the bottom of the stage. The condenser is on top and the light source is also on top. Now this is the image of the inverted microscope. The condenser of the inverted microscope is on top of the stage, and the light source is also on top of the stage. Now this is the eyepiece, So the image is viewed from bottom to top. The image is viewed from bottom to top instead of from top to bottom as it is with other optical microscopes. What are the uses of the inverted microscopes? What are the uses? The inverted microscopes are particularly useful in tissue culture since it allows observation of cells through the bottom of a culture vessel without opening the container and without the air interface normally present between the objective and the surface of the culture. By adding phase contrast optics to the inverted microscope, it is possible to monitor tissue cultures directly without the aid of stains or other enhancements. It's possible to observe tissue cultures directly without the aid of stains and other enhancements. Thanks very much for watching this part of the series. We urge you to continue to subscribe to our YouTube channel to learn more about microscopy. Our next lecture will be on the electron microscopes and many other microscopes. Please subscribe and you'll be glad you did.